Cholesterol is a very important but often misunderstood lipid. It's perhaps one of the more widely known biological molecules because of its importance in nutrition. So we talk about good cholesterol and bad cholesterol, or we talk about diets that are low in cholesterol in order to try to avoid heart problems. Because of these nutritional conversations, it might be easy to think of cholesterol as something bad that we don't want in our body. But in fact, cholesterol and other steroids are a key part of our lipid biomolecules. So they're one of our classes of our different lipids. They have key roles in membrane structure, but also as signaling lipids as well. Cholesterol is just one example of our many different steroid and sterol lipid molecules. Our steroids all share this common fused four-ring carbon structure. So notice our full structure over here on the left. We have three 6-membered rings joined together along with a single 5-membered ring in this typical kind of S-shaped pattern. In order to form a sterol, we'll then add an alcohol group onto our steroid ring structure. So of course that OLS ending of our sterols uses the organic nomenclature to indicate the presence of our alcohol. So our cholesterol, of course, will include that alcohol, and then we'll have various other modifications onto this steroid skeleton in order to create the diverse array of steroids and sterols. Here we can see the full structure for cholesterol. So again, of course, note that steroid nucleus here in the center with our 4 tetracyclic fused rings. We then have an alkyl side chain attached. So we'll often see extra carbon groups attached onto our steroids and sterols in order to make them more nonpolar or hydrophobic. So this is very important for the role of cholesterol in our cell membranes. And then, of course, we have that alcohol group that forms our sterol here, which kind of acts as a polar head group for our cholesterol when it sits in the membrane. The tetracyclic structure of our sterols and steroids ends up being almost planar, so we have a relatively flat structure overall for cholesterol. And that planar structure is just because of the fused nature of our rings. So it's not aromatic like our DNA, so it's not truly planar, but it does end up as a relatively planar structure. The nutritional impact of cholesterol primarily comes from its role in lipoprotein particles that help to carry fats around our body. These lipoprotein particles consist of a core that's filled with our nonpolar lipids that's surrounded by a layer of membrane lipids. So it's a phospholipid monolayer where we just have a single layer coating this kind of fat droplet in the middle, along with a variety of different proteins. We call these apolipoproteins that are bound to this phospholipid monolayer as well. Our cholesterol will sit within our phospholipid monolayer to help make up the structure of our particle, and we'll also have cholesterol esters within our lipid droplet here as well. So cholesterol esters have the alcohol group modified to create an additional alkyl chain in order to make it more hydrophobic to sit in our nonpolar lipid droplet here in the center of our particle. There are 4 different major types of lipoprotein particles that are named based off of their relative density. Each different lipoprotein particle will have varying composition between our more nonpolar fats versus our membrane lipids and our proteins. Now, as we rank our lipoprotein particles from low density to high density, we have our chylomicrons and VLDL, or very low density lipoprotein particles, the low end of the density range. And you see that they have relatively low amounts of proteins and very high percentages of our fats, or our triacylglycerides. We then have low-density lipoprotein particles or high-density lipoprotein particles. You can notice the increase in density here. And we have increasing percentages of proteins and phospholipids and decreasing amounts of our triacylglycerides. We also have much higher concentrations of cholesterol, whether it's in our cholesterol ester form or in our free cholesterol form in our higher density lipoprotein particles, with our LDL containing the highest percentage of cholesterol of all of our particles. The density difference between these particles is also reflected in their size. So our chylomicrons is one of our lower density particles, are the largest. VLDL particles are also quite large, whereas our LDL and HDL particles are much smaller, right? So they have less of those low density fats, and they're more, much more of those higher density proteins and cholesterol. While all of these lipoprotein particles are generally involved in carrying lipids around our body, their specific roles will vary. So that's what will lead to our LDL being known as bad cholesterol, and our HDL is known as good cholesterol. LDL and HDL are specifically involved in transport of cholesterol and other fats both from and to the liver. So LDL will be carrying cholesterol and fats out to other tissues, whether it's to use those fats for energy or to store that in our long-term fat storage. Now, the reason why LDL is sometimes called bad cholesterol is because if it has excess cholesterol and fats there, more than our tissues need, we can end up depositing some of that extra cholesterol and fat in order to form plaques within our arteries. So that, of course, can lead to blockage and can create heart problems over time. On the other hand, HDL is transporting cholesterol back to our liver. And so that cholesterol can even come from these plaques that we have building up in our arteries. And so HDL is often referred to as good cholesterol. So that cholesterol then from our HDL would go back to the liver, where it's then used to produce other compounds, such as our bile acids, which we'll be talking about in a little bit. So already we start to see the complexity of our cholesterol, right? So it can be a bad thing if it's deposited in our arteries, but of course it is a critical component. And if we are able to take it back to our liver and use it for producing other compounds, we're able to derive a useful benefit from that cholesterol. Bile acids are a variety of sterols that are produced from cholesterol. So they are acidic sterols, meaning that they're going to be modified to create acidic functional groups. So here we have cholic acid on the left. So notice we have a carboxylic acid attached onto the alkyl side chain of our cholesterol. We also have some additional alcohol functional groups attached to our steroid nucleus. Over on the right, we have a taurocholic acid, which is one of the most common bile acids. So we have our acidic taurine group attached here with the negatively charged sulfate, in addition to those hydroxy groups that were added. For all of these different bile acids, you should notice that we have very polar groups attached to our nonpolar structure. So all of our groups circled will tend to be relatively polar, whereas, of course, our steroid nucleus is always very nonpolar. The additional polarity of our bile acids enables them to act as detergents within our digestive system. So our bile acids will be released into our intestines in order to help to digest fats. So as our fats are going through our digestive system, they'll form flat globules, or they'll be clumping together due to the hydrophobic effect. And our bile salts will act as a detergent to help to break up these fats into smaller droplets through the formation of an emulsion. So they're essentially forming little micelles of bile salts combined with our fat molecules in the center to help to break those up, where they can then be digested by lipases. So lipases will cut up the individual fats to form free fatty acids, and these can then be taken up into our epithelial cells for eventual digestion and the metabolism of those lipids. So those will actually form into our chylomicron lipoprotein particles, where they'll then be transported to the liver and transported to the rest of our body. Cholesterol is also used to synthesize a variety of different steroid hormones. So this would be the more signaling side of cholesterol and steroids. So steroid hormones are all different varieties of oxidized forms of sterols. So they would be more polar than cholesterol due to our additional oxidized groups. So you can notice we have additional alcohols added on many of these different steroids. We have our sterol here oxidized into a carbonyl for most of these different steroid hormones. Notice we do have a hydroxy group down here for our estradiol. And so overall, they'll tend to be much more polar because of these different polar groups that are added on. So these will be synthesized from cholesterol in gonads and in adrenal glands. There will be relatively small amounts of these hormones produced because they are powerful signaling molecules. So it's not going to require a large amount of cholesterol, but cholesterol will be a key precursor for these hormones. The mixed polar and nonpolar nature of our steroid hormones actually makes them highly effective as signaling molecules. So they are somewhat able to move through polar environments, but they can also move through nonpolar environments. So they will tend to be transported through the bloodstream just because our blood is very polar. But once they are released at their target cells, they'll be able to pass straight into our cell due to their hydrophobic nature. So they can mix with our plasma membrane, but they can also pass through the more polar cytosol as well. So they'll be able to go directly to our nucleus through diffusion, where they'll then target a variety of nuclear receptors to then alter transcription. So these will bind to our DNA and alter the copying of our DNA to create our mRNA transcript. The mRNA, of course, will then lead to changes in cell behavior through the eventual translation and production of new proteins to alter cell function. Again, this whole process is only possible because that hormone is relatively comfortable both within the polar environment of our cell as well as the nonpolar environment of our membranes. Many of our other signaling molecules will actually bind directly to receptors in the cell membrane, as they are much more polar molecules, and we'll have to go through signaling cascades in order to enact changes in our cell behavior. So hormones are able to have a little bit more of a direct impact because of that unique structure. So far, we've looked at the role of cholesterol in nutrition, as well as its role as a signaling molecule through the production of cholesterol-derived steroid hormones. In addition to these roles, cholesterol also plays a key role in our membranes. Almost all cell membranes will contain some variety of sterol-like structures. Eukaryotic membranes will contain different kinds of sterols. So we have cholesterol, whereas other organisms will have other varieties of sterols. So we have phytosterols in plants or ergosterol in fungi. So notice with our ergosterol here, we still share essentially the same structure for most of it, but then our alkyl chain here at the end is modified compared to cholesterol. For other prokaryotic organisms, we won't have normal sterols, but we'll still have very similar looking structure. So we see these hopanols that have this sort of fused cyclic ring system, but then have these alcohols attached on our alkyl chain here. Again, these sterols are able to sit in the membrane because of their shared hydrophobic and polar nature. So, of course, our alcohol groups on each of these sterols and our hopanol will create a more polar end of our lipid molecule, whereas that steroid ring and alkyl chain create a more hydrophobic portion that will sit in our membrane core. So cholesterol will sit right alongside our other phospholipids within that membrane bilayer. The presence of this cholesterol embedded in our membrane helps to increase the overall strength and rigidity of our membrane in order to create the resilient cell membrane structure that we need. So if you have a lipid bilayer that's composed only of phospholipids, you can have two different possibilities. You can either have your phospholipids packed very tightly together in a gel-like solid ordered state. So this would commonly occur if you have a lot of saturated phospholipids that have very high melting points. Or you can melt that bilayer to form a more fluid and disordered membrane where our lipids are more liquid and are able to move around more within that membrane. This solid state is too rigid and inflexible and would not work well as a cell membrane, whereas our liquid state is too disorganized and too flexible. So in order to get around this, we add cholesterol. When we combine cholesterol with our liquid state, we get a more liquid ordered state that's not fully gel, but it's not fully fluid like our state over here on the right. So this is because of the rigid structure of our cholesterol. It kind of interacts with our lipids to help to increase that ordering here within our structure. If we then add our unsaturated phospholipids, which tend to create more disorder because of the bent shape of those unsaturated fatty acid chains, we create this fluid liquid disordered state where we have a membrane that's not quite as flexible as this full fluid state up here, but has a little bit of rigidity from our cholesterol interacting with those saturated phospholipids. So the overall effect is that we're able to increase our membrane rigidity slightly to increase the overall strength of our membrane. So it has the right balance of flexibility and strength. Because our cholesterol likes to clump together with those saturated phospholipids, we actually won't have an even distribution of our cholesterol within the membrane. So it's been proposed that cholesterol will clump together with other saturated phospholipids to form what we call lipid rafts. So these are regions that are enriched in these saturated sphingolipids as well as our cholesterol, and may even have specific proteins bound in those regions as well. So we'll have an asymmetry both within the distribution of a single leaflet, as well as, of course, between our inner and outer leaflets, as we've seen before. Now, it is still uncertain how much this sort of raft formation affects our cell's behavior. So there are some studies that suggest that this raft formation can alter cell signaling, while other studies have suggested that these rafts don't really form any large structures in our membranes and won't have any significant effect on our cell behavior. So it's still an active area of research, but it is quite clear that our lipids will have some organization within a leaflet and won't just be randomly distributed. Because cholesterol impacts the rigidity of our membrane, it will also have a key impact on the permeability of our membrane as well. Permeability, of course, refers to the ability of molecules to move through our membrane. So our membrane is what's known as semi-permeable, which means that it's able to block larger and more charged or polar molecules, while smaller uncharged or more nonpolar molecules are able to pass through the membrane. So depending on that balance of polarity versus hydrophobicity, molecules will be able to slowly pass through our membrane over time. So as time passes, of course, our molecules will want to diffuse through the membrane to even out the concentration on either side of our membrane. From a cellular standpoint, this permeability can be both beneficial and detrimental. So, of course, for some molecules, such as glucose, it's very useful if glucose can diffuse into our cells so we can pick up that extra nutrient. However, it's also very important that we don't have glucose diffusing out of our cells. So, of course, that's why we have phosphate modification of sugars to help lock those sugars inside of our cells. It's also important that we don't have toxic molecules diffusing into our cells as well. right? So you really have to have a balance of a membrane that's not too permeable to just let everything through, but also not so impermeable that we can't get things like key nutrients coming inside of our cells. So cholesterol actually has a key role in lipid permeability. So because it increases the rigidity and the packing of our lipids within the membrane, that will reduce our membrane permeability, right? So as we add in cholesterol here, that cholesterol, again, will help to get our lipids more ordered so that they sit more nicely next to each other, and our membrane becomes more tightly packed. And so then it's harder for our molecules to kind of push their way through those lipids and get into our cell. So as you increase the amount of cholesterol, you'll see that tighter packing and less molecules diffusing in. As you reduce the amount of cholesterol, our membrane again becomes more fluid, and you have more openings where our molecules can diffuse through the membrane. So as we see, there are 3 different key roles of cholesterol within our body. It helps to transport lipids around our body as part of those lipoprotein particles. It acts as a source for a variety of different signaling hormones. And it acts as a key component of our membrane structure. So when you hear somebody say that cholesterol is bad, or you need to lower the amount of cholesterol in your diet, what they're really saying is just that we need a balanced amount of cholesterol, right? If we have too much cholesterol, then that's where it can end up getting deposited in our blood vessels and creating those plaques and those heart problems. If we have too little cholesterol, though, that would also cause problems because it is a critical component of our membranes and a critical source of those signaling lipids. But normally our body, of course, is very good at producing the right amount of cholesterol in order to maintain these key structures. And so if you have very high amounts of cholesterol in your diet, it tends to just produce an excess of cholesterol overall in your body. As we look at the structure and behavior of our cholesterol, we see that it's always critical for that cholesterol to be interacting with other molecules in order to carry out its various functions, right? So when it's transporting lipids, it's working as part of this lipoprotein particle with other proteins that are involved in that particle structure. When it's in our membrane, of course, it's interacting with other phospholipids in order to create that rigid membrane structure. It's often easy in biochemistry to get focused on just one individual molecule and how its structure or function affects its behavior. But it's important to realize that we're always surrounded by all kinds of different molecules, whether it's other lipids or whether it's other biomolecules such as our proteins or our sugars or even our nucleic acids. The membrane is actually a great example of this. So here you can see the fluid mosaic model of our membrane, where the membrane is essentially a sea of lipids with these individual proteins interspersed throughout that membrane. So we can't really think of our membrane as just being lipids by themselves. We have proteins involved there, and those proteins will have various sugars attached as well. So we have our glycoconjugate glycolipids or glycoproteins, and all of these are kind of working together as this complex unit in order to make our membrane function. The same principle applies throughout our cells, right? We don't really have one biomolecule here or one biomolecule there. It's all of our biomolecules working in concert to create this beautiful symphony, which is our cell.